Dear Ada, thank you very much for your presentation. We really enjoyed it very, very much. So we are, unfortunately, we cannot have you here currently personally, but thank you for sharing your very valuable time with us during the discussion. So
it's unfortunate that I cannot be with you. I'm also not very happy about that. But maybe in the future we'll meet person to person.
Thank you very much indeed, Ada. So the students are really active, plenty of questions coming. And the first question would be about the reason is for the existence of natural antibiotics. How do you think this reason can be used in designing new antibiotics?
All the antibiotics that, sorry, not all, except for 2 antibiotics that target ribosomes are natural. The companies just made them more efficient, less poisoning, quicker, more active. So this is the basis of what is going on now. Only 1 or 2 antibiotics, that ribosomal antibiotics, that are from scratch. But I'm not sure that there is a lot of possibilities to use these ribosomal antibiotics, the natural ones. Nature fought much more than we can, and produce much more imaginative antibiotics. Maybe they can still be improved. If somebody wants to try it, I bless them, but I'm not sure.
Okay, thank you. And another question is coming that do you think that developing new small molecular antibiotics can solve the antibiotic resistance crisis? What do you think about the phage therapy, antimicrobial peptides?
I'm not an expert in this. I know that there is some effort, and I wish very good results with it. But to say that I understand it, it's not correct. It's not my expertise. So I wish the best, but I don't know.
One of the students has a nice question about, and I really curiosity whether how to answer it. What do you think was the most important biological experiment of the last 100 years?
During the last 100 years,
Yes.
of most. This is a question that, in principle, I don't like to answer, because I don't like the word most. What look most best today can be just second good tomorrow. It means what is exciting when it comes can be overexciting in the short or long future. So also I cannot answer what is the most complicated or the most difficult, because things are changing. But if we look at the progress of biology, biochemistry, medicine in the last 100 years, it's a huge step forward. And there is a good reason to expect even bigger one, especially now that the structures can be determined much faster without crystals, and because intelligence, artificial intelligence showed that they can recognize paths and symbols within structures, so they can predict many structures, and therefore we can try to understand how these proteins that they predict, how they function, and if they are connected to diseases, we may be able to design new medications. So there are many many, but I cannot. But I think that the fact that we can understand function of biological molecules and complexes and so on, that's a big achievement.
Yes, and the question is continuing about this artificial intelligence-based protein structure determination. So how can you compare it with the pre-bio X-ray crystallography?
It's based on X-ray crystallography. It's based on structures. The patterns that artificial intelligence recognize and use for prediction came from known structure. What's exciting about it, it's above the expectations and very above us, is that the artificial intelligence could identify features better than human beings, much better. Means the prediction level until I think 4 years ago was about around 60%. Earlier it started by 20 or 30%, and now it's 95%, almost full.
Yeah, and the next question is coming about different disaster of the earth. Actually, because if there are too many antibiotics resistance, which can have a big problem. And what do you think in the next 10, 15 years, the pollution or the global warming or the multi-resistance bacterium or maybe viruses, as we can see in COVID-19, will be the biggest problem?
I'm sorry that I won't answer this one. I'm not looking for disasters. I'm looking for positive influence, positive contributions. So if I'm not sure that I even understand what it means, global warming or global pollution. I know we have to look for clean world. I know that we have to make sure that we are not polluting it, but to call disasters, it's beyond me.
Okay, so there is another questions coming. Why didn't the mechanism of cap-independent initiation disappeared in case of eukaryote during evolution in order to make the expression of viral genes impossible?
Can you repeat? I didn't hear well.
Yeah, the question is about the cap-independent initiation disappeared in case of eukaryotes during evolution in order to make the expression of viral gene impossible. So why didn't the mechanism existed?
I don't know. I just don't know. Why didn't it exist? I don't know. I think that we are living quite well. That eukaryotes are living quite well, and this is consequence of evolution from prokaryotes. But to talk about mechanism, this one or that one, it's difficult. Although we think that we saw how prebiotic world became biotic, how the RNA-based world became protein RNA, but this is not answering the question. It's something that we found, and we are working to prove it.
Yes, and some questions coming concerning the scientific grants. Actually, we all know that in plenty of times, if you write the preliminary experiments and everything, and your study is not much risky, you can get sometimes easier a grant application than if you write very risky projects. Sometimes you ended in nowhere and you cannot finish the project. So what do you think, how we can make scientists more interested in a risky project like you did in the past?
And the grants have to be, the philosophy of grants, in my opinion, has to be modified substantially. It doesn't have to be for short time. It doesn't have to count. See what they do now, they count how many papers, how many people read it and that's it. That's not important. What's important is the subject. Sometimes the subject is difficult, but very exciting. It takes years until there is a good paper. And these people hardly can get funds, so they stop this. And the young people are actually thinking more about how to publish fast and how to make it attractive than how to make good science. So I think the funding philosophy has to go back to what it was before, although it was not perfect, but a little bit better. And the universities or institutes have to let people work according to their curiosity rather than according to the number of papers.
Yes, and you mentioned last time, I remember that also in your career, your institution, about your promotion was difficult, if I remember well, because of the risky projects or not publishing enough.
That's correct. It was really difficult. I was fired. I was afterwards hired on lower level. The judging committee, the reviewers that included Sir John Kendrew and Chris Amphisen, Nobel Prize winners, they said that a project like mine shouldn't be stopped, but the chances that I will get to the structure are not sure. Nevertheless, they suggested not to stop, and this is why I was able to continue working, but it was not easy.
Okay, so thank you. Some questions coming about ribosomes. That do you think there are things still left to research about ribosomes? I think the answer is yes, but can you tell a couple of future perspectives on research activities on ribosomes?
So if we started with medicine, so I will continue on this line. There are diseases that are linked to mutations in the genes of ribosome proteins. Very, very severe diseases. Many types of cancers, more than a quarter of the cancers, anemia, and several similar. So research on this, in my opinion, is really needed. That's one thing. Second thing, ribosomes can be a good tool to understand evolution, to understand interactions between proteins and RNA, RNA and proteins. There are some that are trying to produce better ribosomes in the lab that will make unknown, so far unknown compounds like proteins that have the amino acids or have specific functions. So I think there is still a lot to study.
And the next question is, what is your prediction concerning the antibiotic resistance? That can we make antibiotics which can stop the development of resistance, or we will always need to make new type of antibiotics?
You see, the bacteria are very clever. The pathogens are very clever. So whatever we will make, they will find a way to resist it. The question is how long it takes, and if meanwhile we can do other things. So the antibiotics that we suggest that were in the lecture, we have now 25 types of them. So if all of them are good, which we didn't show yet, but let's say 16 are good, and we go one by one, and each of them takes between 10 to 20 years to produce resistance, we have 160 years. Between 160 to 320 years of still a line of new antibiotics. Meanwhile, maybe we will become even more clever. But I think this is quite a nice plan for, not for stopping resistance. We won't be able to do it because bacteria are clever. They want to live, they will find way. but because we can bypass it from the side one by one.
Yes, the next question is coming concerning also about the publications. Around 50 years ago, there were annually about 80,000 publications, which were quite big numbers. Nowadays, last year, 1.4 million publications came out as research activity. Of course, nobody can follow the research phase. So how you select about the articles you would like to read, because of course we don't have time to read many.
We never could read all. And when I was young, there were much less articles, but we needed to see the real journal physically. So it took a lot of time just to get after the journal and open it. Now we just click and we get it on the screen. We will never be able to read all, and even if we read all, we will never be able to understand all. So I think everybody should, or I do, I look for those that in their title there is a word that is of interest for me.
Yes,
even though I don't read all of them. Even
sure.
then I select.
Sure. The next question coming about your big enthusiasms and motivations. So what you decided to keep you ongoing when people were skeptical about your success. So what was your inner motivation?
First of all, I was also skeptical. Every step we made was difficult, and we had to either create new procedures or to bypass it and so on. What kept me? My curiosity. I really wanted to understand how the genetic code is being translated fast and without mistakes, and in all cells.
Some questions also coming, students usually ask about work-life balance. So what do you think about this phrase about work-life balance, or how it worked in your career?
I don't like the word balance, because balance means that you weigh 6 hours here, 6 hours there, one day here, one day there. I hate it, and I never worked according to this. My word, how did I go on? With love. I loved my science, I loved my family, loved my life. So whatever I loved and I had to do, I did, and I didn't complain and I didn't count. I just did it.
So another easy questions coming about what do you think is the key to success?
What is the what?
What is the key? What is the key to be successful, to have success? What is the most important issue?
Curiosity and passion to work. That's all. Everything else comes within it. Passion, love to work, and high level of curiosity.
If you choose a scientific field right now, you would be young. What kind of scientific way or what kind of research you would start?
Whatever is interesting and for me and still is not answered. And I think for everybody should be something like this. And I cannot tell anybody else what will be interesting for her or for him, for anybody.
Yes, we also know that the young scientific education for young people in Israel is absolutely in top. As far as I heard, sometimes they start at nursery. So what do you think?
Sometimes they start what?
Start at nursery in the kindergarten.
How do they start in the kindergarten?
Yeah. So what do you think?
What do they start?
the science education, scientific activities for people. So when do you think the best is to start with young people, talented young people, some science education?
I think that encouraging curiosity is the most important thing. And in Israel, it's not a crime to ask questions that the teacher doesn't know to answer. That's okay. And this is in my opinion, this is the key. The kids that study in kindergarten or in the first years, they study science, it's a low level, but it encourages curiosity. In my opinion, every mother can increase the scientific attitude of their children even when they're young. It means when a child comes back from school, even when very young, usually the parents, the mother asks, what did the teacher say? What did he think? What is the grade? And things like this. And I'm encouraging parents to ask, what did you ask the children? What did you ask the teacher? What did you want him to explain you? So this gives the child the encouragement to ask more and to be more curious.
Yes, the other questions about coming from your life, and also what has the Nobel Prize allowed you to do what you would not be able to do otherwise in the past? So did it change your life or possibilities for the future?
Well, of course it changed. But the best thing that happened to me because of the Nobel is the possibility to interact with students and young people like I'm doing now. Before I got the Nobel, nobody knew about me. Nobody asked me to talk to young people. So the best thing that happened, many things happened, but this is the best. Second thing that happened is media that always needs something, and I don't like it. But the interaction with young people, this is a present, a nice surprise.
Okay, there is a question coming concerning how did you get information that you received the Nobel Prize? So, and did you believe that, or you waited that you in some point you will get the Nobel Prize?
I got it by phone, and I didn't believe. I just said, "Are you making fun of me?" I didn't expect. I did not work for prizes all my life.
So, and is there anything you would like to tell to the students right now about research activities or any kind of things you would like to pass them?
I want to wish them the best, to wish that they love what they do, that they love science, that they love their experiments, and that they keep being curious until even after they solve the first and second and third problems that they were interested in. And I wish that life of scientists will be less automatic, but that's not the young people. These are the funding and the universities, authorities that have to change.
So thank you very much, Ada. And I also saw publications from you, even from the last year and also this year. So you never can stop. And what we can wish that please never stop your research activity and also being with young people. It was a pleasure being with you. And we would like to wish you all the best. And please be away from the new coronavirus. Thank you very much being with us.
Thank you. And thank all of you. And good luck and best wishes to everybody. Bye.
Thank you very much. Bye. And now this part of the program is finished. And however, today you will be able to be with us longer. So please look at the program and come back after the break.
I'm really sorry, but I have previous commitment, so I will leave you now with all my best wishes. Bye.
Bye.